I am David Blumenthal from the Chair of Experimental Bioinformatics of the Technical University of Munich. Today I am presenting EpiGEN, an epistasis simulation pipeline. This is joint work with Lorenzo Viola, Markus List, Jan Baumbach, Paolo Thierry and Tim Kacprowski. Before getting into the details of EpiGEN, let us briefly recap some important concepts. In genome-wide association studies, we would like to link genetic variants to phenotypic traits of interest. Single nucleotide polymorphisms, or short SNPs, are single positions in the genome where two variants, a major and a minor allele, are observed in a population. Now, the problem is that individual SNPs often only explain a fraction of the estimated heritability. One possible reason for this is that SNPs are involved in epistatic interactions. That is, there might be SNPs which are jointly but not individually predictive of the phenotype of interest. So the goal is to develop scalable tools which are capable of detecting biologically relevant cases of epistasis. And for evaluating these tools, we need flexible epistasis simulators that generate realistic data. Here is an overview of EpiGEN's main features. EpiGEN can generate arbitrary phenotypes. It can simulate observation bias, it generates realistic LD patterns, it supports the specification of minor allele frequencies, it can simulate higher order interactions, it allows arbitrary epistasis models, it requires no proprietary software, and it requires no third-party input files. As you can see, EpiGEN stands out from existing simulators in that it combines all of these desirable features. EpiGEN is implemented as a simulation pipeline which consists of four main steps. In the first step, we call HapGen2 to generate the genotype corpora. Note that HapGen2 could in principle be replaced by any other genotype generator that yields realistic LD patterns. Also, EpiGEN is shipped with pre-computed corpora, so the user does not have to call HapGen2 herself. Once that's done, we subsample the SNPs. This can be done either randomly or based on the minor allele frequencies of the noise SNPs and or of the epistasis SNPs. After that, we generate the phenotypes. For this, we use so-called epistasis models, which can either be specified extensionally or via parametrized risk models. The parametrized models support dichotomous and quantitative phenotypes. The extensional models support all kinds of phenotypes. Finally, we subsample the individuals. This can be done randomly or based on a biased target distribution, which models observation bias. For defining epistasis models, we need to introduce some notation. P is the set of all possible phenotypes. So in the quantitative case, P is the set of all reals. In the categorical or dichotomous case, P is the set from 1 up to C, where C is the number of categories. Next, we consider the space D of P of all suitable phenotype distributions. In the quantitative case, D of P is the space of all normal distributions. In the categorical or dichotomous case, D of P is the space of all categorical distributions with C categories. An epistasis model for an interaction of order D is now simply a function M which maps a genotype vector of size D to a suitable phenotype distribution. This phenotype distribution is then used to generate the phenotypes. Hence, epistasis models generalize penetrance tables which are often used to model epistatic interactions. In EpiGEN, the user can provide extensionally defined epistasis models via ini files. For quantitative and dichotomous phenotypes, the epistasis model can alternatively be specified via parametrized risk models. The risk model for an interaction of order D is a function R which maps a genotype vector of size D to a real number. It specifies the disease risk for each possible genotype at the epistasis SNPs. In the quantitative case, the induced epistasis model is given as the normal distribution with mean R of G and standard deviation sigma, which is provided by the user. In the dichotomous case, the induced epistasis model is the binary distribution with a case probability given as R of G divided by 1 plus R of G. In EpiGEN, the user can combine several parametrized risk models. The following interaction models are available: joint dominant interaction, joint recessive interaction, multiplicative interaction, and exponential interaction. Moreover, EpiGEN provides dominant, recessive, and additive marginal risk models. Further risk models can easily be influenced by the user. Here is an overview of the available parametrized risk models for pairwise interactions. In the multiplicative interaction model, the disease risk increases multiplicatively with the number of minor alleles at the epistasis SNPs. In the exponential interaction model, the increase is exponential. In the joint dominant interaction model, there has to be at least one minor allele at both epistasis SNPs for observing an increased risk alpha. In the joint recessive interaction model, two minor alleles at both epistasis SNPs are needed. Using Apigen is easy. Simply clone the Git repository available at github.com/baumbachlab/Apigen and run the Python scripts in a terminal. If you want to learn more about Apigen, please read our paper, which has recently been published in Bioinformatics. Thank you for attending this talk.